Good morning, students. Yesterday we did the introduction part of our chapter on biomolecules, and during which I told you that biomolecules are those molecules which are found in the living organisms. Okay, and I said that biomolecules are of two types: organic molecules and inorganic molecules. Organic molecules, they contain carbon atoms, whereas inorganic molecules, they contain less number of carbon atoms, or they don't contain carbon atoms at all. Inorganic molecules contains mineral ions and salts and water, whereas organic molecules are composed of carbohydrates, proteins, fats and lipids, and nucleic acids. And I told you that organic molecules can be categorized into two groups based upon their molecular weight, right? Micromolecules and macromolecules. Micromolecules are those molecules which has got molecular weight less than 1000 daltons, whereas macromolecules are those molecules which have got molecular weight more than 1000 daltons. And examples are amino acids, nucleotides, simple sugars, and lipids. They are all micromolecules. They have got molecular weight less than 1000 daltons. They are small molecules, whereas proteins, nucleic acids, and polysaccharides, they are all large molecules. They have got molecular weight more than 1000 daltons. So that was a quick revision of what we studied yesterday. Before I start today's lesson, I want you to look over here. This one, right? We have got two groups of molecules, biomolecules: micromolecules and macromolecules. Micromolecules amino acids, simple sugars, nucleotides, and lipids. Macromolecules proteins, polysaccharides, and nucleic acids. Micromolecules, amino acid, I have drawn a corresponding line over here towards proteins. Simple sugars, I have drawn a line towards polysaccharides. Nucleotides, I have drawn line towards nucleic acids. There is a reason behind that. If you look over here, the proteins, polysaccharides, nucleic acids, they are all macromolecules. So they have got molecular weight more than 1000 daltons. They can go up to 10,000 daltons as well, right? So they are pretty large molecules, biomolecules. Now, proteins, polysaccharides, nucleic acids, they are all polymers. Polymers. What do we mean by polymers? So polymers are those long chain of molecules in which they are made up of repeating units. Proteins, polysaccharides, nucleic acid, repeating units ki so protein repeating unit amino acid. So protein the polymer re quick repeating unit the amino acid chadur. So amino acids, they are monomers of proteins. So proteins are polymers, but amino acids, they are the monomers of proteins. Amino acid, a chain of amino acid will make protein. Amino acid and protein chawar. Same goes for polysaccharide as well. Polysaccharides, they are polymers. They are polymers of simple sugars like monosaccharides. So simple sugars, they are monomers of polysaccharides. Yeah, the nucleic acids, DNA, the RNA, right? DNA, RNA, they are nucleic acids and they are all polymers. They are made up of repeating units, and their repeating units are nucleotides. So nucleotides, they are monomers of nucleic acid. Nucleotide mangbo chik thu niro ani nucleic acid chawar. DNA, RNA so wala khar guwar ngazula nucleic acid and nucleotide guwar. Se uchung so polysaccharide so wala ngazula khar guwar monosaccharide mangbo shuj kor. So di indu zambala amino acid da protein dewa doa. Amino acid ki protein suyudwa. Simple sugar ki polysaccharide suyudwa. Kunyi ni para dewa doa. Nucleotides ki nucleic acid suyudwa. Kunyi ni para dewa doa. Adesia. Lipids, they are not, they cannot form polymers. So lipids we will study separately under micromolecules. Amino acid loga jangre, protein jangre jela. But what I will do is, I will teach amino acid. We will discuss about amino acid first, and then we will continue discussing about proteins. Amino acid amino acid ani protein and continuous ani sugar carbohydrate simple sugar jang simple sugar gocho da lamsang khari jangna degree polysaccharide jangna degree kuni ni loga majang wache nyamdo jang shejo. So it will be flow do shile word din nanglo jangdu samba. Okay, so concept ki flow jo mare word takatur ta thangbo ngazu kharne go zu re labu imbaina amino acid. So today in this particular class we will discuss about amino acids and how a chain of amino acids will form proteins. So this we will study in today's class. Amino acids are monomers of proteins. Amino acid the protein gi monomers res, protein the polymer relaro. That is here. That is the tenga doji remove kiranzo de sigdo. The tenga do remove the karitsyongore protein jigsyongor. Tadina siwe mena ribu mangubedo ribu dige sama khajido chain doji form chain ni tenga chagdo. So dinangshi di gangzang kisi protein jigi imba na di ribu dige sama karitsyongor lena they are all amino acids. Dige sama kharre amino acids re. Tadela reme siwe mena dela Histidine, serine, Leucine, Phenylalanine and sangma del doa. So these are all amino acids. The amino acids kharcharu amino acid chigijel chig thuni and kharcharu protein charu do. Adi galangazu jela yangya tagda. Ine so kharu gochu gore amino acid mangbu chig line digchero chigijel chig thuni kharcharu gore protein charu. So amino acids they are monomers of protein. The amino acids amino acids they are organic compounds which contains an amino group, an acidic group, a hydrogen group and a variable group which is designated as R group as a substituent on the same carbon, that is alpha carbon. Carbon chigigala. On the same carbon we have how many groups? Amino group, amino group. We have got acidic group, COOH group, carboxyl group, COOH is an acidic group, right? And then we have a hydrogen, the hydrogen the dedwa, and then we have a variable group designated as R group chi dedwa. Carbon chi gigang la yoba sonza, kunzu shija, the group shi di kharga la thu dedo, carbon chi gigang la thu dedo. So carbon di kharchare, it is the alpha carbon, or alpha carbon gala thu der. So that is why it is called as alpha amino acid. Kisi amino acid structure thila when the remudi kiran jangu gorda. You have to know this, right? You have to practice drawing this diagram, right? Carbon chig gangla group shi khare re amino group, hydrogen chig, carboxyl group chig, and R group chig. R group di jela khari nahi siri yani ye jela lagrwa. R group is a variable group. It can be anything, right? So this here, amino acid is nothing but substituted methanes. Methane lana khare CH4 imba da kena nyama nya jang yosha zigorda methane lawi mena CH4 methane is a CH4 ko structure khare carbon chigi gala hydrogen shi yo ta hydrogen shi na lo ne hydrogen sum di khonzu gi replace chewe imba na substitute chewe imba na substitute chewe na jo jawala oro hydrogen chigi sabla amino group chig jar hydrogen d gi sabla carboxyl group chig jar ani hydrogen d gi sabla R group chig jar wa imba na ani ko khar chadu re amino acid chadu re slar. So they are substituted methanes. Amino acids they are nothing but substituted methanes, right? Kisi amino acid giteshe kangza ma jago they are organic compounds which contains amino group, an acidic group, a hydrogen group, and a R group on the same carbon, attached to the same carbon. So that is called as amino acid. So amino acid ki structure diche. Amino acid ki structure di abun jayen da de slide din nalone. Ani kalakazam ju gochu sar kesi dhara structure de gochu beim by na tadesia based on the nature of R group. Tadi R group the kharila variable relasa ko jodo digrela so there are many types of amino acids. So amino acid rig manda mangu pe resra rig manda mangu dige sama khari gi chasu gora dige sumja rangsa digda amino group, carboxyl group, hydrogen group the rangsa digre R group the change hasuna amino acid rig change hadu or so there are different types of amino groups, amino acids based upon R group kana shishane. Amino acid rig mangbu yore tenro. Protein soyala amino acid khazu yore. There are 20 types of amino acids. So there are 20 types of amino acids which makes up the proteins found in living organisms. Living organisms nalo yogi protein soyala amino acid khazu yore lana kha manda khazu yore nishu thamba yor. So the example khashishiteo. Kisi R group gi sabla hydrogen chi gyo bi mana the R group di sabla kirangi hydrogen jona ko variable ro so R di sabla hydrogen chi gyo bi mana ko khar cha dure glycine cha dure sheba samar anja de or the NH2 di de do COOH di de do hydrogen di de dangja de do so R gi sabla the box di nawla R gi sabla hydrogen him bai na ko khar cha dure glycine che mingla kisi R gi sabla methyl group jo bi mana CH3 jo bi bai na ko khar cha dure alanine ro kisi R group gi sabla tela methyl hydroxy methyl geo be imba na ko khar chadu re serine chadu CH2OH geo be imba na So R group gala shishani ani amino acid rig mata to. So ngazu khen tomne total khajo yorlas amino acid nishu thamba ngazu gi subu nanglo la organism dige gi subu nanglo la protein soya chu the amino acid rig mata khajo yore nishu thamba yores ra ta nishu thamba ne example sum dete so konjo khandis khewa khandis tu gorla na R group gala khewa tu gor R group the H in a glycine R group the CH3 in a alanine R group the CH2OH in a serine and dinangshi R group mish gala shishani ani ngazu lo ta sa nishu thamba yor se ushon so. So we have now 20 different types of amino acids inside the organism's body to make proteins. Proteins so amino acid rig manta nishu yores ro. Konso sangma kharila khewa durla na R functional group kala khewa durla ro. Ta amino acid kha manta nishu dila physical properties yore, chemical properties yore. Sangma ga ji physical property the chemical property chikpa chagurla na chikpa chagmar kon sangma amino acid loga ro. D in the zambala konso ki physical the chemical property the kharikala shisha wore. How does an amino acid get a physical and a chemical property? They are based upon the groups amino group, carboxyl group, R functional group. Konjo ki group dige sum gala shishani ani amino acid la physical the chemical property manda yor. Tapena cha shawa imbana some of the amino acids they are acidic in nature. Konjo ki property khari re chemical property chi khari acidic nature chadur. Penchashana glutamic acid, aspartic acid. So these two amino acids, kuni amino acid da glutamic acid, the aspartic acid la sto, kuni amino acid re kuni gi nature khare la na acidic nature re. Athokol acidic property dikhandes le re la wa yimba na ani desida COO group the dependent di glutamic acid ro glutamate lane degree glutamic acid lane degree ro aspartic acid lane degree aspartate lane degree adi kharaz motu achi mato. So glutamic acid ki structure C wa yimba na glutamic acid nanglo la alpha carbon gala COOH chikdwa din shi R group nanglo le COO group chidwa dela di imba sonza ko kharcha du gore acidic cha du gore COOH group din ye khare la so acidic group re la so di imba sonza ko kharcha du gore acidic cha du gore ta dela R group din nanglo khardo carboxyl group chik thewas to di imba sonza ko kharcha du gore acidic cha du ta desia some of the amino acids they are basic in nature. Amino acid kashe khare basic re basic property or base ki property or penachana lysine, arginine, histidine do dinzo. Tang azu lysine ki structure la C wi imba na lysine la khari thewa do amino group jik thewa do. Alpha carbon gala amino group jik do yoyo bache. Dinashi R group gala amino group jik thewa sto. Di masonza amino acid dekho kharcha do re basic in nature ta dis konyo bu do amino acid la ni mingta yore ne ko basic re la no khala gore samejo. So amino acid la dila basic nature ge yong si da lysine the arginine the histidine dige sama khare basic amino acid re ro so ko basic kharchin re la mino khola amino group di thewa yor desi da NH2 group chik dele chik doa ani R group gale NH2 group chik doa di basong ko kharcha do re base chadur se shon so and some of them they are neutral Valine kesi amino group da carboxyl group ni de manyu chik ba yo be na ani amino acid khorang kharcha do re neutral chadur acid ge maimba base ge maimba. Okay, so they are neither acid nor base, so they are neutral amino acids like valine. Now some of the amino acids they are aromatic amino acids. The aromatic amino acids lana khala or example tyrosine, phenylalanine, tryptophan lanitedwa. So these are examples of aromatic amino acids. Aromatic amino acids are those amino acids in which the R group contains an aromatic ring. Tela benzene ring does to aromatic ring yo be imbana ani amino acid dikhar chadu re aromatic amino acids chadu. So property jo marwe amino acid gi khoranzola physical property the chemical property di kharigala ra ligre lana ko group denzo gala ra ligre amino group carboxyl group and R functional group gala ra ligre lar. So again I said there are 20 different types of amino acids to make protein within an organism's body. Organism ki subu nanglola protein soela amino acid rigmanta khajola so nishu thamba yores ta de nishu thamba ga list te yorda ta nishu di manju gi ya category ni na lo kopshe gir khari gala shisha ni kopshe gire ro kisi kira ngobuji kopshe gire na chik shisha sa roj goro on what basis are we categorizing them into these two groups? Right, non-essential amino acids. There are 11 non-essential amino acids and there are 9 essential amino acids. So they are categorized based upon whether our body can make these amino acids on our own or not. Kisi ngaju subu chi gi roa, ngaju subu gi amino acid chik sue thugu yobi imbai na. If our body can synthesize an amino acid, they are called as non-essential amino acid. If our body can synthesize an amino acid, that amino acid is called as non-essential amino acid. So these are all the amino acids that our body can make. Ngazu subugi amino acid dige samo suetuor. So therefore they are said non-essential. 9 essential amino acids to histidine, isoleucine dash to dige samo ngauto ju marda. These 9 amino acids, they cannot be made by our body. Synthesized. These amino acids, they cannot be synthesized by our body. Therefore, they are essential amino acids. Non-essential, the essential. These amino acids, they are said to be essential amino acids because they cannot be made in our bodies. Therefore, it is essential in our diet. Amino acids that are essential in our diet, they are said to be essential amino acids. Tadi yabu ko so khala na le inji miji gugor khala na le inji miji yo gongejire essential jigre khala na inji miji khajin yo gugore ngazu subu so to go mar khala ne mara we imana amino acid li marwa subu na lola. So khala na lola go yo gyoshto dezola ngazu khala gore essential amino acid khala na la khajin gugore because our body cannot make it. So non-essential amino acid khala na le inji miji go mar, they are not required in our diet. Why? Because our body can make it on our own. Essential amino acid chigro khala nal khajin go mare lawa imba na alanine khajin khala nal go mara ngazu khala saya dina alanine khajin go marla na maranzu subur cell gi alanine swetu yor synthesize chetu yor di imba sonza ani alanine di kharchare non essential amino acid char. So now one particular property of amino acid is the ionizable nature of NH2 and COOH group. Amino acid nanglo khare carbon chigala amino group da carboxyl group jigoro and NH2 da COOH group dioro kunyi ni khare ionizable lana kunyi ni kala charge layer ro NH2 di NH3 chayer and NH3 positive chagre COOH di COOO chayer COO ro hydrogen di large and ko kharcha dure negative chadur. So charge change chagdo NH3 chani positive chagre COO chani ko kharcha dure negative chadur. So ionizable nature do the group ni di imba sonza amino acid raw. Hence in solution of different pH the structure of amino acid it also changes. Kisi rangi amino acid chik hiro amino acid chik solution na lu ba imba na solution digi pH kan la shishani amino acid la structure manda yor seo chungse raw. For example that the wapar dina tan pe seo chagre kisi nganzu gi acidic solution jinalo amino acid lu ba imba na ta hypothetically da acidic solution jinanglo la a dirda acidic solution jinanglo la amino acid jilu bi na the structure kharcha do NH2 the ionized chani ra NH2 the ionized chani NH3 cha so positive charge le so overall ko la charge khar resha positive charge lig do equals so silita du zambala if we put the amino acid in basic solution tadi acidic solution ra sujo dikhare basic solution na lu ba na ra strong base na lu we na COOH mara group di ko gane hydrogen dillani COO negative chagdo. So overall charge khar chagdo amino acid ki overall charge ti negatively charged redo. Acidic solution na lola positively charged, basic solution na lola negatively charged. These are the kima de chigyor te sida kima jela isoelectric point la di tadi neutral rela wo marda tadi kena sem na yabu nyamado this does not mean this particular amino acid is in neutral pH. Neutral pH la yor lawo marda. So this is at a particular pH point in which now see over here both NH2 as well as COOH they are both ionized. Niga ionized chadir. NH2 the NH3 positive chadir, COOH the COO negative chadir. Niga positive. So overall charge khar chagdo 0. Positive ge yore negative yor kunyi cancel out chaduro. So overall charge khar chadure 0 chadur neutral chadur. Seo chungse dela positive charge dela negative charge dela 0 charge che yomar. Seo chungso. So di lang azu khar lawore this structure is called as Zwitterion. This particular structure is called as Zwitterion. That is same na yabu nyamato. This is called as Zwitterion. That di any point dila khalawore at the particular point in the pH that particular pH point at which amino acid will occur as a Zwitterion, that is called as isoelectric point. Isoelectric point la imena pH tang azu gi chigne chini chungni phodo yo imena ro pH khwala ani amino acid di Zwitterion chagdo ro ko the negative ge mai ba positive ge mai ba khadu chagdo di ha ko ena di point dila azu khala gore isoelectric point lagre seo chungso and the structure dila khala lagre Zwitterion lagre. 세우 총수에 디 property 디 간데스 리고 어레라고 임바나 NH2 다 COOH 니디 ionize 차요 NH2 this NH3 chani positive charge over COOH the COO chani negative charge over. So they get charged. That is called as ionization. Okay. So this is deuterionic form. So this is all about the properties of amino acids. Amino acid lana amino acid ki structure amino acid la rig manda ani amino acid ki property khashish ba acidic in acidic khachin negative basic in the basic kharchin re aromatic lana aromatic kharchin re dige sama ba and then last nga jo khar deuterionic form di amino acid can occur in amino acid can occur as a deuterion at a particular pH point known as isoelectric point, during which the amino acid is neither positively charged nor negatively charged. It is neutral. Okay, so that was all about amino acids. The next proteins. Protein is a macromolecule, right? But it is made up of amino acids. So just now we studied about amino acids. Now we will follow it up with discussing upon proteins. So proteins ki khari ki dubsha yore amino acid ki dubsha yoro. Amino acid jang sasre dijela khari jangna yabo proteins kana jangna yabo. So tomorrow we will study about proteins. Now look over here. You just read section 9.4 and 9.7 as an assignment for today. Thari kiranzo section 9.4 the 9.7 di mallo da. 9.4 the kiranzo proteins lan title do. 9.7 the title kharasha structure of proteins. So tomorrow we will discuss about those two topics. But before we discuss tomorrow, you have to read your textbook. Okay. So section 9.4, 9.7 the law. Okay. Yeah.